Good morning, students. So today class 11 biology. Today we are going to discuss practicals of biology class 11 and the experiment number is 5. Today we are going to discuss experiment number practical number 5 of class biology. So what the practical? The practical is number 5 is study of specimen and identification through permanent slides or with the help of museum specimens. So we have to study we have discussed the 5 kingdom classification. We have discussed the 5 kingdoms: Monera, Protista, Fungi, Plantae, Animalia. Okay. So the specimen, the organism of what? Belonging to bacteria, Monera, Protista, Fungi, Plantae, Animalia, we have to study by seeing either seeing the slide, permanent slides, or by seeing the museum specimen. Today I am going to discuss the specimen with the help of slides. Okay? So what we are going to discuss today? Today we are going to discuss study of the slide of bacteria, Lactobacillus, the bacterium causing curdling of milk, the study of slide of Cyanobacteria oscillatoria, the study of slide of Cyanobacteria non-stuck, the study of slide of green alga Spirogyra, and study of slide of the fungus yeast. You know, yeast is a kind of unicellular fungus and yeast multiplies by budding. So budding yeast, structure of yeast and multiplication in yeast by budding we are going to discuss today. First of all, what things we require? First of all, our requirements are slides, the permanent slides. I have placed the slides over here. These are the slides which we are going to discuss today, and this is the microscope, compound microscope we need. So we have to focus the slide, we have to place the slide on this microscope. Suppose anyone I want to study his cell. So this is the slide of his cell, and towards the window side, the direction of light, this microscope has to be placed. And then we have to set the slide, place the slide onto the stage, fix it with the help of clip, focus the mirror, adjust the mirror, and then now light is coming, light is falling on the specimen or slide. Then we can adjust the slide with the help of broad adjustment screw. We can set the slide with the help of broad screw, and once it has been set by the broad screw, then we can adjust with the help of fine screw. Then we use the fine screw to adjust this. So broad screw has been, with the help of broad screw we have set the slide, and now we can use the fine screw for the fine tuning or adjustment. So now the slide has been set. Now we can see the what each cell we can see. We can see the oval cells, each cell unicellular yeast, and few cells are multiplying, are arranged in chain forming the bud, showing the budding. So this way we have to see the slides. Okay, one by one. So the teacher may set the slide, or you may be asked to set the slide. You try your best to set the slide. If you are not able to set the slide, then teacher will set the slide, and you have to observe the slide carefully. You have to see the slide carefully. You have to draw the rough diagram in the classes, but this is the classroom. Now during the lockdown period, this is the classroom. So perhaps you are not, won't come to the school this session. So everything will be finished through this process, online classes only, practical theory both. So this is the procedure. What I have to do, what you have to do, that I have discussed. Now let me come to the topic. First of all, we have to see the slide of bacteria, a typical bacterial cell. A typical bacterial cell. Okay? So the slide we are having, slide of what? Curve. By using the curve, a permanent slide has been made, and that slide is there, and that slide represents Lactobacillus. Lactobacillus, this is a kind of bacterium, a rod-shaped bacterium. This rod-shaped bacterium, and this causes curdling of milk. So lactobacillus, it is rod shaped. When you see the slide, when you focus the slide, you will find many very fine rod-like structures present on it in the slide. Fine, many, not 2, 3, 4. There are many fine rod-like structures like this. So these are the lactobacillus. Now the next slide is of BGA. Blue-green algae bacteria belong to Monera. BGA also belong to Monera. They don't possess what? Nucleus and membrane-bound cell organelles. Now the next slide is of BGA, and BGA we are going to discuss the slide of Oscillatoria. Oscillatoria is a kind of filamentous multicellular BGA. Blue-green algae, cyanobacteria, its body structure is filamentous and it is multicellular, filamentous. Okay, so this is the uppermost cell is what? Dome shaped. Multicellular. Covered by, filament is covered by mucilaginous sheath. Mucilage sheath. Okay, this prevents desiccation or drying up. This is aquatic, found in the aquatic medium. So, no, it is not green, but you cannot see the chloroplast. It is green, it can carry out photosynthesis. A kind of BGA called Oscillatoria. It is filamentous and it shows oscillatory movement. It shows oscillatory movement like this kind of movement. Hence the name Oscillatoria. It oscillates like this. Hence the name Oscillatoria. Kind of BGA. Next one is another type of BGA, nostoc. Nostoc is another kind of BGA, blue-green algae or cyanobacteria. It is filamentous. It is also filamentous and embedded into a gelatinous mass or sheath. This is the jelly-like sheath. In jelly-like sheath, many filaments are embedded in the jelly-like sheath. I have shown the one filament. One filament has been taken out and has been magnified. So this is the one filament. There are many such filaments inside. So this mucilaginous sheath prevents desiccation or drying up. So this is the single filament of the nostoc. It is made of what? Oval cells, oval or rounded cells. These are vegetative cells. Here and there you can find some large cells, some larger cells larger than other cells. These larger cells, double walled, thick walled, called heterocyst. Heterocyst lacks chlorophyll. इसमें chlorophyll pigment नहीं होता है. Other cells possess chlorophyll pigments. Heterocysts do not possess chlorophyll pigments. So heterocysts are concerned with nitrogen fixation. So this is the filament of nostoc. This is nostoc. Now let me come to the green alga. Green alga, Spirogyra. Now the next slide is Spirogyra. When you focus the slide, or I will focus the slide, and when you are asked to see the slide, you will find a structure like this. A filamentous structure you will find. Okay, it is also multicellular. Made of what? Many cells. It is multicellular filament-like structure. It is lush green, shining green, and this is commonly called pond shell or pond esker, found in fresh water, running water. A special point, chloroplast. Cells are uninucleated. These are the nucleus. One nucleus, one cell, one nucleus. Okay, and chloroplast. What is the shape of the chloroplast? The chloroplast is ribbon shaped. Chloroplast is ribbon-like. So ribbon-shaped chloroplast is the special feature of the Spirogyra. Okay. So reproduction, vegetative भी हो सकता है by fragmentation, and sexual reproduction it shows by conjugation. It reproduces sexually by conjugation and asexually by vegetatively by fragmentation. The filaments can fragment and can give rise to new spirogyra. Simple. The filament is attached to the surface or any structure or body with the help of holdfast, and it is what? Silky. Its filaments are long and lush green, shining green and silky, found in fresh water, running water. So these three we have discussed. Lactobacillus. 4 we have discussed. Slide 4 slides we have discussed. Lactobacillus, Oscillatoria, Nostoc, ठीक है, Nostoc and Spirogyra. One more I would like to discuss the slide of. So how to write? We have to write the comments, name of the experiment, and then we have to write numbering A, B, C, D, or 1, 2, 3 like that. Experiment number 5, A, B, C, D. A, you have to write this way. You have to see the slide, and after seeing the slide, you have to write the comments. You have to write the comments, few lines comments, and then draw the diagram on the white sheet. Comments and diagram on the comments on the root sheet and diagram on the white sheet. Labeled diagram should be there. For each and every slide or specimen, you have to write the comment on root sheet and diagram on white sheet. Okay? Now students, we are going to discuss the fifth specimen, study of yeast showing budding. Yeast cell showing budding. You know, yeast is a kind of fungus. You know that yeast is a kind of fungus, and yeast is a what? A unicellular fungus. Yeast is unicellular. Okay? You see the cell. This is the single cell of the yeast. Nucleus, single cell. So it is a fungus. So its cell wall is there, and the cell wall is made of chitin or fungus cellulose. Now you can see over here. This is the what? Budding. The budding is taking place in the yeast. Now this mature cell forms the bud, outgrowth called bud. This nucleus divides into two. One will remain in this cell, and the second nucleus will migrate into the bud. So this way it may form one bud, or it may form chain of buds. So this is chain of buds. So this is the phenomenon called torulation. इसको torulation कहते हैं. So now this torulation, due to torulation, chain of buds, now it becomes multicellular. It appears multicellular. Okay. So question is there: is this a unicellular or fungus? Then why it has been placed in fungi? Why not in protista? Because of this reason only. It is unicellular. Basically, it is unicellular. It reproduces, multiplies by budding. But sometimes due to budding, what happens? Birds fail to segregate. Birds fail to separate out, and chain of birds develop. When chain of birds is formed, in this case it becomes multicellular, and that's why if it develops multicellular stage, then it should be in kingdom fungi, not in protista. Got the point? So now the class is Ascomycetes. It is a kind of fungus, unicellular fungus. Unicellular, and it belongs to class Ascomycetes. A very simple structure. You have to draw the diagram, and you can see the cell. You can set the slide, and in the slide you will find a structure like this. You will see in the slide. Okay. Now, how to write the practical? How to write the practical of the study of a specimen? So first of all, you have to write the features, general features, characteristics. You have to write. You have to write the position also, to which group, which kingdom does it belong to. ठीक है, for example, Nostoc, it belongs to BGA. It is a kind of BGA, filamentous है, it belongs to BGA. ठीक है, and kingdom Monera, and what kingdom Monera? It is a kind of BGA cyanobacteria. Kingdom Monera or cyanobacteria. So BGA belongs to what? Monera. Prokaryotes, Prokaryota, BGA or cyanobacteria, commonly called blue green algae. So this is the identification. So after writing the features, the characteristics of features, ultimately, lastly you have to identify what is it. So identification should be at last, and you have to write the kingdom or class or phylum of the specimen you have to mention. For example, yeast. You have to study yeast. You have to write the comment, and after writing the comments, you have to write, at last you have to write, yeast is what? Unicellular fungus. It what? It belongs to kingdom fungi and class Ascomycetes. Scientific name also you have to write. is scientific name Saccharomyces cerevisiae. So this way you have to finish. This way you have to write prepare the practical file. So you must buy the practical record book, as well as practical manual. In practical manual, everything is given. From there you can write. Don't copy it. Write in past tense passive voice and write a brief note only. Write the very important points only. Write 2, 3, 4 points of identification and then identify the specimen, draw the diagram and label it. And you must write the date of the experiment also. On the drawing sheet, comment sheet, drawing sheet, as well as in the index also, you have to make a list, and in that list also you have to put the date of the experiment. Okay, thank you. That's all.